Hello, my name is Aseel. I'm a PhD candidate at the Perkins Lab at U Ottawa, and today I'm going to explain my research on cell type-specific DNA signatures of transcription factor binding. As we know, we have multiple transcription factors that can bind to multiple regions along the genome and different cell types, and the difference in binding may be due to the intrinsic binding behavior of the transcription factor in question, or it could be due to other mechanisms such as chromatin accessibility, competitive binding, and alternative splicing. And these mechanisms could result in a DNA signature of differential binding. Now, we use the term DNA signature to encompass the different mechanisms that could lead to the differential binding preference of the transcription factor in question. To study the cell type-specific DNA signature of transcription factors, we propose a two-step deep learning method called signatures of transcription factor binding to detect and quantify cell type specificity in a transcription factor's DNA binding signature. We conduct a wide-scale investigation of 194 transcription factors assayed by one or more antibodies across various cell types. That gives us a total of 230 distinct transcription factor and antibody pairs. And then to further explain the biology behind the cell type specificity, we conduct a wide-scale motif enrichment analysis of the transcription factors in question. Here we present a simplified diagram of our approach where we have stage 1 in the red box here, where stage 1 takes as input the DNA sequence of our ChIP-seq peak and outputs whether or not that ChIP-seq peak or that sequence is bound or unbound across various cell types. In stage 2, in addition to this DNA sequence, we include the cell type general, cell type specific concatenation or information. So the cell type general information is a vector of zeros where we're not informing the model about the cell type, where with the cell type specific, we are informing the model about the cell type, and it is the one-hot encoding of the cell type in question. And the idea is to use this information to be able to predict whether or not the sequence is bound or unbound in that specific cell type. The difference in performance between cell type general, cell type specific cases could give us an indication of the cell type specificity of the transcription factors in question. We train a model for each transcription factor and antibody pair. We then obtain the cell type specificity of the various 230 transcription factor and antibody models. And here in the scatter plot, we have the cell type specific AUC, where we're informing the model about the cell type in question. Then we have the cell type general AUC, where we're not informing the model about the cell type in question. And each point in the scatter plot corresponds to a transcription factor and antibody pair. We have a continuum of cell type specificity, so across the diagonal we have transcription factors with lower degrees of cell type specificity, meaning that actually giving the model the cell type specific information does not improve learning. However, in the upper left corner we have transcription factors with a higher degree of cell type specificity, indicating that giving the model information about cell type does indeed improve learning here. We then look at the distribution of the AUC differences in this histogram here, where we highlight the bins in red that contain these specific transcription factors. We notice that transcription factors associated with cancer, such as MYC, BASH1, and ATF7, have a higher degree of cell type specificity, unlike those such as CTCF and HCFC1, which are more associated with the general cell type processes. We then obtain or look at the consistency and performance across antibodies for a specific transcription factor in question. And the idea is, will we have consistency in cell type specificity across the different antibodies for a specific transcription factor? Here we have 24 transcription factors, which have more than one antibody assayed. And along the line for each transcription factor, the different experiments corresponding to the different antibodies. We notice that there is consistency in AUC difference across most transcription factors, such as CTCF. However, for some cases, such as ETV6, we notice there is a lack of consistency where we have actually a big jump in AUC difference from 0 to 0.37. It would be interesting to further investigate this. This could be because of the difference in the antibody used, or maybe the difference in the dataset quality, or maybe the difference in the actual cell types we're looking at as well. Next, we look at motif enrichment. More specifically, we look at the motif patterns that are driving the cell type specific behavior of certain transcription factors and the lack of in other transcription factors as well. So we investigate the motif enrichment more specifically here for ATF7, which is known to have a higher degree of cell type specificity. We apply in silico mutagenesis on the test sequences of ATF7, and we notice that for the specific test sequence here, which is considered to be a peak and is a peak in HepG2 and MCF7, and considered not to be a peak and is not a peak in GM128 and KF562, and also with the concatenation of all zeros, it is not predicted as a peak. We notice that for these two cell types, HepG2 and MCF7, the addition of cell type specific information does indeed improve learning and help identify these specific motif patterns, which correspond to the ATF2 motif pattern. We apply this to all the test sequences and obtain this heat map here by extracting subsequences from these sequences that have the greatest impact on prediction, and then search these subsequences with known Jasper motifs using FEMO. This is the ratio of the number of significant motifs from the bound peaks for each cell type. And we notice that we have two main groupings here. So for the HepG2 and MCF7, we notice similar motif patterns across, or motif enrichment levels across the different motifs. For K562 and GM2078, we notice that we have a different motifs enriched for these two cell types. For example, for the ATF2, ATF3, ATF4, although we know that we see that it's enriched in all four cell types, we notice that it's more enriched in HepG2 and MCF7. However, not as much as in K562 and GM1278. Conversely, we notice that SP1, for example, is more enriched in K562, a little bit more enriched GM1278, and it's not enriched at all in HepG2 and MCF7. So we notice two groupings here in the motif enrichment heat map. We also obtain the gene expression of the motifs in these different cell types, and we notice that for some cases, such as ATF2, CTCFL, we notice similar patterns of motif enrichment and gene expression. However, this is not the case for all motifs, where we notice dissimilar motif enrichment and gene expression patterns, such as for the KL4 case here, and this would be interesting to actually further investigate later on. We also investigate a transcription factor that has a lower degree of cell type specificity, as we saw in our histogram above. Here we obtain the heat map for CTCF across 14 cell types, and we notice more or less similar motif enrichment levels across the different motifs and different cell types, where for the CTCFL has a higher degree of motif enrichment across all the cell types. However, it is consistent. And that further supports our conclusion that CTCF has a lower degree of cell type specificity. We also obtain the gene expression patterns for the motifs here. We then examine motif enrichment across various transcription factor antibody combinations. As previously discussed, we apply the in silico mutagenesis and FEMO on all the transcription factor antibody models here. More specifically, we display those that are part of transcription factor families that are known to have identical DNA binding domains. We have here in the heat map, the columns correspond to the transcription factor antibody and cell type combinations, while the rows correspond to the motifs enriched in the specific ChIP-seq experiments. And each transcription factor family has a different unique color to indicate that these experiments are part of this specific transcription factor family. We see clusterings where we have similar motif patterns or motif enrichment levels in transcription factors from the same transcription factor family. However, we do observe also, once we zoom in onto these clusters, that there are varying degrees of cell type specificity across cell types for a specific transcription factor. We finally provide a graphical representation of the most significant motif hits in 3 cell types, which is the K562, which corresponds to leukemia, the A549, which corresponds to lung cancer, and the HepG2, which corresponds to liver cancer. And we display the motif networks here, where each node corresponds to either a transcription factor or a motif, and the directed arrows point from transcription factors that are part of ChIP-seq experiments to the motifs that are enriched in these ChIP-seq experiments. So the different colors of the arrows indicate whether or not it's enriched in one cell type only, two cell types, or all cell types. For example, the dashed maroon indicates that it's enriched in all cell types, so such as here for GAPA, the ChIP-seq experiment with GAPA, we notice that all these motifs are enriched in all three cancerous cell lines, which makes sense as the ETS transcription factor family is usually associated with cancerous cell lines. We then look at another grouping here, which is the AP1 transcription factor family, which contains subfamilies of FOS, MAF, JUN, and ATF. We notice that for JUN-D, for example, most of the AP1 transcription factors are enriched in JUN-D in all three cancerous cell types. However, as AP1 transcription factors are sometimes oncogenes in cell types, and tumor suppressors and others, we notice here, for example, that for A549 and for HepG2, we notice that the JUN is the ATF or ATF7 or the AP1 transcription factors are enriched in JUN. However, for K562, we do not notice that it's enriched there. So here we observe cell type specific behavior of the transcription factor JUN in the different cell types. So in conclusion, we demonstrate the existence of cell type specificity in 30% of the transcription factors and support our results using motif enrichment analysis. Our comprehensive investigation provides a basis for further study of the mechanisms behind differences in transcription factor DNA binding in different cell types. So this is the end of my poster presentation. If for more information, please contact me at this email, and thank you so much for listening.